Hello again and welcome to Chapter 10, Photosynthesis, where we will talk about the process of converting carbon dioxide and water with the power of light into primarily sugar and oxygen. By now you should have completed the worksheet on photosynthesis that I assigned to you. And hopefully that will give you a good introduction into this chapter, which generally is a tougher chapter for students because they haven't had very much of it. So my goal in this chapter is a different approach. Give you a little bit of material up front in the interactive video, and then have you learn the details that we want to get you commensurate with this class. So we are going to drill a little bit deeper than the HHMI, but if you followed the HHMI and you completed the questions, you've already got a good start into the material. Recall from our bioenergetics discussion is that we have to have a sort of energy flow. There has to be an energy input, a transformation, and then eventually an energy output. The inputs in our ecosystem include sunlight, and that's where photosynthesis plays, but they can also include molecules that have been created by other processes, including radioactivity or gravity and pressure. But those latter two are much rarer in our ecosystem than sunlight is, and that's what makes photosynthesis so preeminently important, is that it is the conduit to energy into our system. Organisms which can utilize non-biological energy sources are what we call autotrophs. They sustain themselves without having to eat any other biologic organisms. They can use nutrients that are essentially inorganic, sunlight or molecules from deep within the earth that have formed through pressure and or radioactive processes. So autotrophs are producers fundamentally. That is to say, and maybe a better word is converters, because they are converting inorganic sources of energy into organic molecules. That's what we mean by production, is that we're producing organic molecules, and they're going to do this using CO2 and other inorganic sources. Heterotrophs, on the other hand, depend on biological energy sources. Photoautotrophs are the ones that we're going to be talking about in this chapter. These are the ones using energy from sunlight to make those organic molecules and nutrients that heterotrophs can then consume and use for their energy sources. Photosynthesis occurring in different kinds of organisms, not just plants. We can see them in algae, certain animal-like cells we call protists, and some prokaryotic cells. For instance, cyanobacteria, for instance, is photosynthetic, but it is not a eukaryote. But they are energy converters. They're converting energy not only to feed themselves, but they usually convert enough that the energy can be supplied to all the other heterotrophs, of which there are legions throughout the world. As you hopefully learned from the video, we have light-dependent reactions, reactions that create NADPH and ATP using light-dependent processes. Both of those molecules feed what we call the Calvin cycle, which is a cyclic set of reactions, not unlike citric acid cycle, only instead of breaking down more complex molecules into simpler ones, what we're doing is we're taking simple molecules and using NADPH and ATP to make more complex molecules. So we have two photosystems, photosystem 1 and photosystem 2. The first photosystem we encounter is actually photosystem 2, and that's in between photosystem 1 and 2 is where we generate most of our ATP. We're going to do that using chemiosmosis. As we bump up electrons using photosystem 2, those electrons lose energy through electron transport, and that's where we pump protons like we did in the mitochondria. Only instead of the mitochondria, we're going to use the thylakoid membrane to hold on to that gradient of protons. And just like in in the mitochondria, we let those protons come out through the ATP synthase to make ATP. So right here we have a high concentration of protons. They are diffusing down their gradient through the ATP synthase, this turbine-like system, and we are forcing phosphate onto ADP in the process. Calvin's cycle, on the other hand, uses NADPH and ATP, but it's what we call the light-independent reactions. It can occur absent of light and often does in order to conserve water in some cases, but it's cyclic just like the citric acid cycle, and we're making more complex molecules. Now it says glucose here, but that's not technically correct. We're actually making a smaller 3-carbon molecule called G3P. And I've already mentioned heterotrophs, so I won't beat this too up, but they obtain their energy and organic material from organic sources. That is to say, they convert the energy and molecules from autotrophs into organic molecules and or waste products in the process of doing work and so forth. Almost all heterotrophs depend on photoautotrophs. There are some very, very rare exceptions. There are animals that live in and around these volcanic vents, and what they do is they feed on bacteria that are autotrophs from the volcanic vents, and so they are heterotrophs to the bacteria that are subsisting on the energy from the force of gravity and radioactive processes deep inside the earth that generate heat and create high energy molecules. It's just a dramatic example of that. The bacteria that are here on in the colors of green and yellow and orange and then eventually brown out here are all feeding off of the energy that is supplied from this hot spring. The molecules, the sulfates, the high energy compounds that are bubbling up through this hot spring are then supplying the energy for these bacteria. So they are feeding on inorganic energy sources and nutrients. That said, this spring is also an ecosystem unto itself. There are certainly bacteria living off of the molecules coming from the hot spring, but there are also organisms like a certain species of fly that feed on the bacteria. So we have a micro ecosystem here. In fact, these flies have to stay within about 3 or 4 inches of the hot spring all their lives, because during the winter it gets so very cold. If they are more than a few seconds away from the energy source, they'll freeze to death. So they have to stay very, very close to the energy source in order to survive. But we have autotrophs in this beautiful colors that we see, not the blues, but certainly the greens, the yellows, and the oranges, and the browns. And then we have organisms that feed on the bacteria. But the vast majority of energy, with probably the exception of nuclear energy, comes from the sun in one way or another. For instance, many, many years ago, you would have peat bogs that had lots and lots of organic matter in them that eventually got buried and then pressed by the Earth's gravity into what we now know as coal beds. That said, buried peat is still used for energy in certain parts of the world. I was just giving a lecture the other day in my FYS class about Ireland. And in Ireland, there aren't enough trees to supply the need for fires and cooking and so forth in the homes in Ireland. And so many homes will burn buried peat like firewood. It hasn't turned quite into coal, but it is still very rich in carbon. And when we have enough heat from an activation energy and oxygen, it can burn just like coal can. But all of our natural resources for energy, gas, oil, and coal, are derived indirectly from sunlight that shone on the Earth millions of years ago. We've talked about chloroplasts in earlier lectures, and we believe that chloroplasts formed because of the endosymbiont hypothesis. That is to say, maybe perhaps a cyanobacterium of some kind was living in very, very close near symbiotic relationships with our eukaryotic precursors, and eventually merged to form a photosynthetic organism. Make sure that you do know all of these terms. It's important that you be able to know all these terms and recognize them, because it will make your life easier when it comes time to answering questions. If you have these terms, it will be easier to answer, because you will then not have to use so many words to describe a process. So understand the structural organization of a chloroplast, and part of the reason for this structure is for regulation and sustainment of photosynthesis. And plants are complex organisms. We don't always give them credit for being as complex as they actually are. In terms of biochemistry, they have humans destroyed. They are so much more complicated biochemically than even the most complex human being. That's because they can't move. They have to solve their homeostatic problems using biochemistry, whereas humans and animals can use behaviors rather than chemistry to solve certain problems. But the green color that we see is actually the reflected light off of the molecules inside the leaves. And so chlorophyll will absorb blue light and red light, but it doesn't really absorb too much green and maybe certain yellows. And that's why the leaves are the color that they are, because the predominant photosynthetic pigment in leaves is chlorophyll, and it happens to reflect green light. Now, not all plant cells are the same, and as you may have learned in our HHMI video, that most of the chloroplasts in a leaf and in plants are found in specialized photosynthetic cells that we call mesophyll or mesophyll cells. They're mostly interior inside the leaf. They're not found very much on the exterior surfaces. That's because they depend on water, and they depend on very, very good regulation of their external environments. So they actually depend on other cells to protect them while they're undergoing photosynthesis. And mesophyll cells contain somewhere between 30 and 40 chloroplasts. There are chloroplasts in lots and lots of cells. They're not only in mesophyll cells, but the mesophyll or mesophyll cells are the ones that have the most chloroplasts. And the arrangement of the cells in a leaf is not unlike the arrangement we have in our own skin. There's an epidermis layer of cells that may be somewhat living, maybe somewhat non-living, more non-living in our situation, but maybe more living in plants. And then you have some, like what they call palisade cells, which are basically like protective layer there to help keep water interior, because we need water for photosynthesis. And then we have that spongy layer, the mesophyll or mesophyll layer. And then we have other structures that resemble like our blood vessels or other subdermal tissue like veins and so forth. And we even have what are called stomata. A single one would be a stoma, and plural would be stomata. And those stomata are surrounded by specialized cells which can contract and relax, called guard cells. So even a relatively simple structure, seemingly like a leaf, is actually a complex mixture of different tissues and different cell types, each with discrete functions. So here's a mesophyll cell, and you can see the chloroplast. And the chloroplast will actually move around in some cases in cells. You can actually see the movement. One of the questions that I typically like to ask is what are some things that are similar to plants and animals and or mitochondria and chloroplasts? Well, one thing that's similar is that both animals and plants have to undergo gas exchange. the case of animals, we have gas exchange occurring at in the lungs or in gills, sometimes through the skin, like in the case of amphibians that live underwater, they can exchange gas through their skin. In the case of a leaf, the gas exchange is occurring in the leaf. It's not occurring hardly anywhere else in the plant. And the leaves use stomata, so they use those pores. The similarity between mitochondria and chloroplasts, there are several things. And again, you should know those similarities versus the differences. Both mitochondria and chloroplasts have a double outer membrane. Actually, that's an outer and an inner membrane. I should be correct on that. It's a double membrane on the outside of the organelle. That said, there's also a membranous interconnected network of sacs. An individual sac is a thylakoid. If we have a stack of those sacs, that would be called a granum, a singular. And if we have multiple stacks, and again, these are interconnected, they would be called grana. There's also a cytosol-like fluid inside chloroplasts, which is similar to mitochondrial matrix, only this time we call it stroma. So it's very, very rich and dense. Lots of nutrients, lots of molecules that we're going to use to create more complex molecules. This is the stroma in chloroplasts, and its matrix in mitochondria. And so here's a nice picture showing a granum here, multiple grana. Again, they're interconnected. An individual thylakoid, and they look like a stack of pancakes. And you can see a really beautiful electron microscope figure of a chloroplast. Now, you can actually track individual atoms through the process of photosynthesis. That is to say, you can look at a carbon here and label it, and that's been done. They're actually able to selectively label carbon with a radioactive tracer, and you can literally follow it through the entire set of reactions, the light-dependent reactions, the shuttling over into the light-independent reactions, the Calvin cycle, and then you can spot those radioactive carbons in the final product of carbohydrate. But it's just the opposite of respiration. The only other ingredient that we have here that we don't have in respiration is light. That's because we're starting with oxidized reactants and we're turning them into reduced products. Okay? We are taking electrons, we're stripping it from the oxygen and the water, and we're putting it into glucose, creating elemental oxygen and a little bit of metabolic water. Sometimes we abbreviate light energy with the Greek letter lambda. So if you see lambda out there, think light energy, think photons of light. In addition to being able to track carbon, you can also track oxygen and even hydrogen if you choose so. Using proper radioactive isotopes of carbon or oxygen or hydrogen, you can follow these atoms through the process, where we start with CO2, oxidized carbon. We start with water, and we input energy to create these reduced products, the glucose, and so forth. And so what we're fundamentally doing in the light-dependent side of the reaction is splitting water. That's really the number one thing that's going on in the light-dependent side of this. The main product here is oxygen, and this is where oxygen comes from photosynthetic autotrophs. Is as we split water, the individual oxygens that are here pair up in order to share their electrons, and that's why we get diatomic oxygen. That's because we're stripping the electrons off and using those electrons to do other things. Like respiration, photosynthesis is a redox process, only we're going in reverse. We're starting with low energy molecules. We're using energy that we're trapping from sunlight in order to create high energy molecules. we are fundamentally reducing carbon dioxide. And that's not reducing carbon dioxide in the atmosphere to say, yes, it's lowering carbon dioxide in the atmosphere too, but that's where it can be a little bit confusing, especially if you go out and try to, look up carbon dioxide reduction. You'll find a lot of Green New Deal stuff. What we're talking about here is the chemical reduction. That's because we're adding electrons to it. We're stripping electrons from water. That's why we're oxidizing. Leo is a German. Lose electrons, oxidation. Gain electrons, reduction. But it is endergonic. It requires energy. It requires a boost of energy in order to create these complex molecules. And again, this is a photo situation, but in other molecules, you can use heat or other chemical sources of energy to do this same thing. It's just that they are not photoautotrophs. And this is just a larger picture from before. Just recall that Leo is a German. We lose electrons, oxidize. We gain electrons, we're reducing. So the two stages of photosynthesis, we have the light reactions and the dark reactions. So the light is the photo part, and the Calvin cycle is the synthesis part. In the light reactions, we're splitting and stripping off electrons off water, and that's why we have oxygen. We are taking those electrons, converting them over to NADP, which makes NADPH, and we're also going to harvest some of that energy to make ATP, what we call photophosphorylation, but it's very, very, very similar to electron transport chain and chemiosmosis that we observe in mitochondria. And like with the citric acid cycle, I want you to learn the general phases of what's going on, but most importantly, I want you to understand what's going on in the very, very first reaction. The enzyme that catalyzes this is called rubisco. So I'm giving that to you right now. You want to make sure you get this first carbon fixation step sorted down. What's the enzyme involved and what is this process? It's called carbon fixation. We're going to use a little bit of ATP and NADPH. That's where the reduction is going on. That's where we're transferring the electrons. And one of the molecules that we create in one cycle of this Calvin cycle is a sugar. It's a 3-carbon sugar called G3P. Okay, G3P, that's the abbreviation. It's a 3-carbon sugar. Now what's left over, we recycle. So we're going to regenerate. So we bring in some carbon, we add some electrons, we harvest that reduced carbon, and we recycle using ATP the molecule so that we can fix more carbon. That's the Calvin cycle in a nutshell. But get to know this very, very first reaction, the one that's catalyzed by rubisco. But the 3 phases, carbon fixation, reduction where the electrons are inserted, and then we're going to harvest some sugar, and then we're going to regenerate the molecule so that it can accept new carbons. Here are the inputs for the light reactions. Certainly sunlight, you have that letter lambda, water, and that's one of the reasons why mesophyll cells are the ones that do most of this, because you need to have a pretty good supply of water to do this. If you don't have water, you can't do photosynthesis. And we have the precursors, NADP+. This is an oxidizing reagent that we're going to reduce. ADP and some inorganic phosphate. When we're done with the light reactions, we'll have some ATP and we'll have NADPH, which will serve as fuel for the Calvin cycle. Now, we make ATP in mitochondria in plants as well. So this is not the only source of ATP found in a plant cell. That Calvin cycle will eventually regenerate ADP and PI, regenerate oxidized NADP. In the process, we will have harvested some sugar, okay, and we're ready to accept more carbon dioxide in the dark phases. So we're going to start with the light-dependent reactions, the reactions that depend on sunlight. But before we do, we need to talk about sunlight a little bit and talk about how chlorophyll behaves in sunlight. So just a little bit about the nature of sunlight and how it works. The band of wavelengths that we call visible light is actually very, very narrow on the overall spectrum, what we call the electromagnetic spectrum, which is energy that utilizes not only motion, but also involves perpendicular axis of motion in terms of magnetism and electrical current. And visible light's a very, very small piece of that. What you can see is that we have wavelength, and wavelength is proportional to the amount of energy that's in the electromagnetic radiation that we're talking about. The narrower the peaks, the more energy there is. Because light is relatively constant in its speed, if you have less frequency, then the waves are further apart by virtue of the speed of light. And so light travels in those rhythmic waves just like heat does, or radio, or X-rays, or gamma rays. It's the same stuff. It's just a matter of how much energy is present. And so we have to talk about wavelengths because chlorophyll and our instruments that can detect light or transmit light have to be tuned properly to the appropriate wavelength, or the systems don't work. So wavelength is what determines the type of electromagnetic energy. The longer the wavelength, the more like heat and radio it is. The shorter the wavelength of light, the more like gamma radiation or cosmic radiation it is. And the higher the energy. So higher energy, lower energy. You can see the range of wavelengths here and the objects that can be resolved by those wavelengths. So the longer wavelengths can be used to detect things, but they have to be very, very large. Higher energy beams can detect smaller things, but the problem is there's so much energy here as they often will rip apart the object that you're looking at after a very, very short period of time. Even with visible light, some of the molecules that we try to visualize sometimes get photo bleached and destroyed by the energy that we're using to observe them. So visible light's right here, somewhere in the 380 to 780 nanometers, 380 billionths of a meter, 780 billionths of a meter. They behave like waves, but they also behave like particles. And that's the real magic of electromagnetic radiation, is that it can behave like particles and like waves. Another picture showing the long waves on the right. Then we have red, which is at the longest wavelength, purple, which is at the shortest wavelength, and then we have UV. So we have infrared. These are longer than the red that we can see. And we have ultraviolet, which is shorter than the wavelength that we can see. And we go into X-rays and gamma rays and even cosmic rays after that are even higher energy. So what happens is the pigments, that includes chlorophyll and other pigments, and even different flavors of chlorophyll, like chlorophyll A and chlorophyll B and even chlorophyll F, have different frequencies that they can absorb and use to convert into chemical energy. But you can see out here in the middle, right in green, we don't absorb anything, and that's why we see that reflected green. And that's why wavelengths become very, very important. Because if you are in different environmental situations, like for instance, if you're in around a whole bunch of other leaves from other plants, you may have trouble getting enough energy to sustain your photosynthetic activity. And so you have different pigments, and they absorb different wavelengths with different levels of efficiency. The wavelengths that are absorbed, either you're converting the energy to something else, or it's converting the energy to heat. Those are the two things that happen. We either take that energy and convert it into complex organic molecules, or it becomes heat that then has to be dissipated from the leaf itself. And the reason, like I said earlier, that the leaves are green is because we are reflecting most of our light from the green wavelength. Yeah, a little bit of orange, a little bit of yellow, but the truth is we reflect a lot of green, and that's why these things appear as green. And this can actually be measured using techniques that will take white light or any light source that you have. And what you do is you cause that light to separate into discrete frequencies. And if you have a special filter that will only allow a certain frequency through, you can see how much of that energy either gets absorbed by the chlorophyll solution in this case, or doesn't get absorbed and passes through and thus strikes a photoelectric tube. And you can look at how much electricity is produced. If it's not producing very much electricity, that means most of the energy is being absorbed. If it's producing a lot of electricity, that means most of it is not being absorbed. We call this spectroscopy. So if we're shining, let's say, a green wavelength, the green doesn't get absorbed, and so we see a high number on the electric meter. If we were to shine a blue wavelength, which we saw in the previous slide, gets absorbed, most of that energy is absorbed. This would actually start to rise in temperature, for one thing, and you wouldn't see a whole lot of electricity showing up on our photovoltmeter because not a lot of that energy is making it through. It's being absorbed by the chlorophyll. So we're going to stop right there for now. We just hit the 30-minute mark. And next time we'll work a little bit more on the spectrum of chlorophyll, particularly chlorophyll A and B. And then we'll start working further and further into the light-dependent reactions. So thanks for your time, and I will talk to you in the next video. See you soon.